Inheritance of two genes. So today we will discuss about Mendel's dihybrid cross. So we are calling it as a dihybrid cross because in this cross or hybridization, Mendel has taken two contrasting characters. For example, shape of a sheet and color of a sheet. So if we see these two contrasting characters, it will have each characters will have a dominant and recessive forms. So if we see the first character, that is a shape of a seed, so when its allele will be in its dominant form, the shape of the seed will be round, and when the allele of this gene will be in recessive form, it will the shape of the seed will be wrinkled. And if we see the second character, that is the color of a sheet, so when the alleles for this characteristics will be in dominant form, the color of the sheet will be yellow, and when it will be in recessive form, it will be green. So in dihybrid cross, Mendel crossed a pure homozygous round and yellow seeded plant with a pure homozygous wrinkled and green seeded plant. So while doing the crossing, one allele will be received by the gamete for one characteristic. So if we take the pure yellow and round, so for pure yellow only capital Y, and for round characteristics only R allele will be received by a single gamete. So this is also very important point, that for a single characteristics only a single allele will be received by a gamete. So then we will do the crossing or fusion, and after the fusions we will get the members of F1 generation. So in F1 generations, all the individuals had yellow and round sheets. So now next we will do the crossing between the male and female individuals of F1 generation. So after doing the selfing between the individuals of F1 generations, we can get the four type of the combination in F2 generation. So we can get 9 round yellow plants, 3 wrinkled yellow plants, 3 round green plants, and 1 wrinkled green plant. So the ratio will be 9:3:3:1. So the phenotypic ratio in F2 generation in dihybrid cross, it will be 9:3:3:1. And the genotypic ratios of the dihybrid cross will be 1:2:2:2:1:2:1:2:1. So on the basis of these ratio, Mendel's established second law, and that law is called as a law of independent assortment, which is based on the F2 ratio of dihybrid cross. It means that the gene for these two characters do not influence each other. This conclusions promoted Mendel's to formulate his principle of independent assortment. So on the basis of results of dihybrid cross, Mendel established his law, which is called as a Mendel's law of independent assortment. So according to this law, when two pairs of traits are combined in dihybrid, then segregation of one pair of trait is independent of the other pair of trait. It basically states that the genes of one characters do not influence the genes of the another characters in any way. And during interactions, they remain completely independent of each other and assort independent. As Mendel was not aware of linkage, crossing over, and gene interactions. So a later finding reveals out the fact that genes which are present on the same homologous chromosomes show same degree of interactions. Therefore, Mendel's third law or principle is modified as the gene of one character do not influence the gene of another character if they are present on different homologous chromosomes. That means if the genes for the two different genes for the two different characteristics are present on two different homologous chromosomes, then they do not interfere the inheritance of the both gene.